Hello, welcome everybody to the first Exposome Symposium. This is more of a continuation of an initiative that we did in one of the EHEN projects, that's Exemius. We did this frequent topical webinars where we had wonderful speakers share their experience about exposome research, but also a few specific topics on chemical regulation and other aspects of it. Today's symposium is titled Cumulative Exposures, Collective Impact, Advancing Public Health Through Exposome Research. And since this is the first presentation or symposium from Exposome, there are a few housekeeping announcements, but also some information that we would like to share before we start. So the event is being recorded. You will have access to the chat box and the Q&A, so where you can drop your question for the speakers. Each speaker will do their presentation, and then we will have time to interact and ask questions and discuss with them a bit in details. Post symposium, should you have any questions or any other information that you need from us, do write to us. You can find us on our website or social media channels. Here we will be monitoring your the chat box. So Katran and I will be monitoring the chat box as well as the Q&A. Normally your microphone and screen sharing features are disabled, but depending on how the presentation progresses, we might want to have a more active discussion. We will see to it later on. A bit about Exposim. Exposim is a consortium of several partners and coordinated from KU Leuven. I'm one of the co-coordinators together with Professor Peter Hood, and we are a geographically balanced group with experts from epidemiology, immunology, omics, analytics, as well as social sciences and humanities. And this is something that started from, and it's a bit of a bridge from the Exemius project. These are some of the statistics for you, where 70% of EU citizens face several of the urban stressors, including different pollutants, chemicals, noise, airborne allergens, with almost 25% of the European population affected by several of the immune-mediated diseases, and the prevalence is quite high in women for a lot of the autoimmune diseases. So we wanted to address some of these gaps through an integrated approach, where we started with an initial part on the knowledge mapping exercise, which you can see here on the top, using machine learning, text mining exercises, and approaches like adverse outcome pathways and networks, and then validating some of these biomarkers in or biobank samples, as well as setting up cohort studies to come to end results, which can inform policy through toolboxes, policy roadmaps, as well as white papers. We are organized into several work packages, but the knowledge mapping, as I mentioned, includes inventories of existing guidelines and policies across Europe, but also beyond Europe, uh, topical reviews on interventions, as well as modeling of external and internal exposome. A lot of registry-based studies, which includes UK Biobank datasets, previous datasets on lifelines from the Netherlands, but also other birth cohorts. I'm sorry, this is moving too fast, but you have a collection of birth cohorts, including Preventadal and Vironage FIPOS, as well as occupational cohorts, which kind of inform the knowledge translation and policy. It's important to mention that we are just not the only project, but there are 6 other projects as part of the Expo Health Net cluster, which is kind of a bridge between the EHEN, IHEN, and now Expo Health Net, which is continuing the exposome research. There are the projects, and you can find us on the Expo Health Net website, but also recording of our kickoff meeting. You can find a lot of resources in the website, and if you're open to collaboration, please reach out to us. Our scientific strategy as well as policy strategy would be published soon, and so you can find us and get in collaboration for several of your projects. And who better to kick off our symposium than Gary Miller, with so much of experience with Exposomics research. And Gary, I have a book of yours just next to me here. Have read it from front to end of it. So Gary will be talking about Exposomics from wild concept to scientific reality. Gary is Vice Dean, Strategy and Innovation at Columbia University, US, with a lot of experience in exposomics, molecular mechanism linking, environment, and health. So Gary, I give the floor to you, and then I'll moderate the chat as well. Thank you.
Okay. Thank you very much for the invitation. I'm always excited to talk about the exposome, as you know. Let me just get my slides pulled up here. That should be full screen now. Is that good?
Yes.
Yeah, it's good.
Okay.
Thank you.
Okay, so yeah, the title. This is a very meaningful title. You know, we know that Chris Wilde was the one that proposed this concept 20 years ago, a little over 20 years ago now. And, you know, I think we spent a good decade with it in a sort of concept phase. And then over the last, 10 years or so, people have been advancing more of the methods and techniques. And I propose that we really are at this point where we are at the point of it being scientific reality. And I'll give some examples of why I think that's the case. And so again, Chris introduced this concept. He then became the director of IARC for many years. And he defined it as, it encompasses all life course environmental exposures from prenatal period onwards. Some people like to think about it, as everything that's non-genetic. And we've had many different ways of kind of going through and defining it. I think in some ways it's caused a bit of confusion in the field. And I think we've found some ways to address that. But we know that we're looking at these very comprehensive types of exposures. We're talking about what comes from the ecosystem in green space and blue space, lifestyle factors, socioeconomic factors, and then all the sorts of physical and chemical things that we classically think about in, the people that are doing more like toxicology and environmental epidemiology, but we're trying to get all of these things together. And I think what's really critical about this is that when you look at how we study and understand human disease and biology, we think about our genes and our environment. And genetics has just been extraordinarily successful. After the Human Genome Project, you could do genome-wide association studies, and they've done that across almost all diseases. And so we have this really strong analysis of G, but I would argue that the E part has fallen short. And it's just because the methods hadn't been there, but I think now they are. And so this E that I always show in this equation being a little bit smaller than the G, I think it's time for that to have equal footing to the genome. But it's not something to be done in isolation, because it really is the interaction between our genes and environment that really play a key role. And I also think it's very important about, we're not just looking at describing the exposures themselves, because if any of these things are affecting our health, they have to affect our biology. It's not a magical association. It's, there's something that has to happen. Whether or not there's still a signal of it you can measure is a different question. But the idea is that if there's something that's leading to an increased rate of a disease, there has to be a biological underpinning to that. And so we need to study that mechanistic connection the entire time. So again, like I mentioned, some of the things with the definition and some confusion in the field. And so we convened this meeting back in 2023 at Cold Spring Harbor Laboratory at the Banbury Center. And this was at the suggestion of a colleague of mine at Columbia, Tom Maniotis. Anyone who learned how to clone 30 or so years ago read the Maniotis manual. So he spent a long time at Cold Spring Harbor Laboratory and thought this would be a good place to do this. And so we pulled together a couple dozen people from all over different backgrounds and to sit down and really kind of hash out what the definition should be. And there were people from Europe, and probably some people on this call attended. And so the definition we came up with, this integrated compilation of all physical, chemical, biological, and psychosocial influences that impact biology. You can add things onto it, and health, and there's things you can do, but this core definition, I think, is really critical to remind people we're trying to get all of these things integrated together. This paper was published back in April in Science, and I think it's really been helpful. I know from like the U.S. side in our scientific leadership at NIH, having a document they can look at that has a good overview and definition of it has been very helpful in kind of promoting the concept and getting more support. I also think it's still important for us to work on how we define it. And it's critical to do it in different ways in populations because different audiences want different types of information. And so I like to think about it as all of these different things we do, like we measure chemicals and molecules in various biological samples. We can measure drugs and their metabolites. We have personal monitoring, wearable devices, sensors, and then we can link all those things up with geocoding sort of information. It's going to take a lot of advanced analytics and things like AI to pull all this data together and ultimately get into these sort of exposome-wide association studies in very much analogous to what GWAS had been doing. But the idea is that we're going to be using multi-omic laboratory methods to measure as many of these xenobiotics, environmental chemicals, and small molecules as possible. But then also, again, like thinking about epigenetic modifications, RNA expression, changes in protein, we really want to look at all of these things together. Again, using all the geospatial information, residential history to get all that, the historical perspective of it, and to help make decisions on policy. If you know where the source of some of these things are coming from, you can use that in your policy kind of guidance in that way. And again, I think that a key part of this is going to be how I don't think artificial intelligence is magic. I don't think it's just a matter of throwing all the data into it, but we do have many more sophisticated tools now that I think will help us make sense of this data. But it is going to take a lot of work to get that data integration and workflow in a way that's actually coherent and usable. I think this paper here is a really good example of the importance of what we do in exposomics. This came out in Nature Medicine early last year. Austin Argentiera, when he was at Oxford, published this paper where he looked, this was from UK Biobank, and he took all the epidemiological measures they had in there, like 160 different things. He looked at the polygenic risk score and then developed like an exposome risk score of all those different factors. And then he looked at the top causes of death and across each of those diseases, what predicted death for people that had those diseases. And so you can see a big range. The biggest part of it is age and sex. And so like the older you are, and if you're a man for most of these things, it increases the likelihood of you dying. And so age is one of the biggest factors. But when you look at the polygenic risk score, very predictive of many outcomes and diseases. But when you look at all-cause mortality, when you combine them all together, you can see the green part, the exposome part, is a much bigger predictor of mortality than the polygenic risk score. Now, a caveat there is all of these polygenic risk scores are about disease incidence. There's been a lot of talk in genomics about they need to do a better job of like getting a polygenic risk score for disease progression, and they weren't designed to do that. Some of them do, but I think there are some gaps there. But either way, you're never going to overcome this part where the exposome has a big part of it. Now, the one thing, and I talked to Austin and his group quite a bit about this, is this study was done in UK Biobank, and they didn't use any of the biobank samples. Right? The idea is you have blood samples on all of these people, and so you could go through and do the biochemical analysis on it and get much more information on that. And that's important because when you look at the total prediction of this model, they're far away for all-cause mortality to the 1.0. And so there's still a lot more to be explained. And I think that's where the biochemistry can really help do that. But I think it's a very good example of showing how important studying the non-genetic drivers of disease are to really understand, many, many different human diseases. And so this was, one of the big motivations back when we started our Hercules Center in 2013 was to think about the methods we would need to do this sort of analysis. And so working very closely with Dean Jones at Emory, we just kept pushing on the high resolution mass spectrometry, which back in 2013 was just kind of coming along, and really working to make these methods more sensitive, more reliable, get better coverage of chemical space. We had this paper about a year and a half ago in ES&T where we worked with many people across the world, 20 different institutions across 7 countries, looking at all these methods and how we get to the point of harmonizing it and really pushing these things together. But I think, to me, one of the key parts is that we can measure many of these different chemicals. I'm still never concerned when we're in a non-targeted or untargeted format of having unidentified peaks. I want to know what those peaks are, but we don't have to know what they are until we know they're really worth looking at. And I think, to me, that's one of the key parts is like when people talk about most of the peaks are unknown, they're not unknowable. There's just some level of confidence in what they are. We usually have a pretty good clue of what they are. We just don't have that exact match that we want to have that we can get in different situations. And so it's really, I think we've seen such improvement over the last decade. I think we're going to continue to see more. I think there's also some really interesting things when you think about like social determinants of health is we've seen many sorts of chemical exposures that are correlated to like the neighborhood deprivation index or things that are, thought of in the sort of social constructs, but it makes sense because if a person lives in a neighborhood that's close to a factory or a highway, you'd expect them to have higher exposures to various chemicals. And this is something where we can kind of bridge some of these social sort of factors and help identify more of like a molecular mechanism of what might be happening there. So we use these methods going into various human diseases. This was something we did a few years ago, relatively small study initially. Now we were up in the thousands, but we could easily separate cases and controls in Alzheimer's disease and separate the different racial ethnic backgrounds of the participants in the study. And I recall the neurologist I was working with on that, he was surprised because he'd never seen the groups separate that way. And my comment was that, well, it could just be something like diet. And he's like, well, it can't be diet because we did a dietary inventory. And I think we know that these dietary inventories, they're not biochemistry. They're estimates of exposure. Whereas when you're measuring the chemicals in a person's body, if they're using different oils to cook their foods at different temperatures, those sorts of molecules that are formed, like we can detect those things. And so it shows that you can pick up these, nuanced changes there. And this is important because we see across different racial ethnic groups is different vulnerability to disease. And so when you can start seeing, yes, they have a higher incidence in one population, and you see different changes in their biology and their metabolome in this space because we measure all the exogenous and endogenous at the same time, you can start getting clues about how, what might be driving the different conditions. And so I said, we've been able to scale these studies up now and get to the point of doing kind of an exposome-wide, metabolome-wide association study for Alzheimer's disease. And this was a really interesting kind of tale with how we did this work and our methods. We thought we had good methods to measure the various chemicals. And when we initially did the analysis, it was looking at clinical Alzheimer's disease. And we didn't really see many signals there. And it was a bit frustrating because I thought we knew what we were doing and was sure we would see something. But during the course of this project, these new biomarkers for Alzheimer's disease came out. The p-tau217 that's now clinically available, p-tau181. And so during the study, we started getting these biomarkers measured on all the participants. And so we then could go through and use a biomarker-informed diagnosis of AD. And essentially what this is doing is throwing out 15% of the participants who don't have idiopathic Alzheimer's disease. They had maybe vascular dementia or a different part of it because they didn't have this tau pathology. And so when you had that, when you start looking at these biomarkers of disease, you see many more hits coming out here because one, you're measuring these biomarkers of disease in the same blood sample as we're doing all this mass spectrometry on, but that these things are known to track the disease progression. And so you get this better link together. And as you can see, like when you look at the sort of network plots, is that many of these features are correlating with the changes in the biomarkers of the disease in a much stronger way than just the clinical diagnosis of it. And so we're excited about this project. This is all my LC analysis, the gas chromatography, high resolution mass spec. We're analyzing the data right now, but we really saw some strong features in the study. So we did this in this population that was about a third white, a third Caribbean Hispanic, and a third African American in two different populations. And then we wanted to replicate it in a different population, this ROSMAP cohort, where they also have brain pathology on over 100 people. So here, these are white participants, and we'd seen these changes in these lyso-PCs in the previous figure. And so we went through and did it in this other population and saw these same changes across many of these different lysophosphatidylcholines. And these have been implicated previously as having a role in transporting the neuroprotective polyunsaturated fatty acids into the brain. And so we're finding changes in molecules occurring across multiple cohorts. We weren't looking for this, right? We had no a priori hypothesis. This was just looking at what the data were driving through here. And we were able to link these things together with the p-tau and the lyso-PCs, and also then validate it in these different cohorts. And so it just shows that by taking these very broad approaches and not looking under the street lamp, you truly can find novel associations and new confirmation for different sorts of studies that link these things in. And so now I think we're really trying to get now more on that environmental side of it. And like I said, we have much of that data coming along. But one of the examples in the initial study we did, Rinda Kalia and my group had geocoded information on the people and their residential history. And she looked at all these different markers of air pollution, because we had good air pollution maps in New York City, and found that she was able to link the levels of exposure of PM2.5 to alterations in glutathione intermediates in the plasma of the participants. It's like you're getting these very external markers linking down to actually molecular changes that are occurring in people that are involved in redox. Like it was, on the face, it looks very logical to see that sort of connection. And we'll be able to do this on the entire population because we have them all geocoded. And so I think we're really going to make some headway here in connecting with these exposomic approaches for things like Alzheimer's. Also started a collaboration with Roel Vermeulen and Susan Peters in Utrecht on Parkinson's disease. And this was looking at, in the EPIC cohort, there's an EPIC for PD because several hundred people developed Parkinson's after they enrolled in that study back in the 90s. So these were collected over 25 years ago, between 8 and 10 years after they entered the study. That was the average age they developed Parkinson's disease. And so these samples were before the people knew they had the disease. And so we were looking for first, and just kind of like a reality check for us, was that they had asked people about their coffee consumption when they entered the study 25 years ago. And so they had coffee consumption, had it quintiled out based on, they calculated based on the type of coffee and things they drank. And so we just said, well, what if we just measured caffeine and all the caffeine metabolites in them and see if we get a good correspondence with that? And so we did, and it was exactly what we'd expect, is that we saw theophylline, paraxanthine, caffeine, and many different downstream metabolites elevated, they went up perfectly with how much people said they were drinking coffee. And we know coffee's protective in Parkinson's. And so we saw that same connection there. But it shows how you can look at these older biobank samples and find out things about lifestyle factors that occurred many, many years before. We also saw some very interesting things with microbial metabolites. I don't really have time to go into that, but again, we get very broad coverage of chemical space. And we could also measure a lot of pesticides. Like, so when you think about looking at a few different pesticides linking it to Parkinson's, we were able to measure over 300 pesticide molecules in this population of just a few hundred people. The one thing I think we did see is, like, while we saw some associations with some of these triazines, I don't think the study population was really, don't have quite the power. I think many more of these would have come up had we had a larger population. And I think that just kind of makes it this point, is that we do need to think about how we've learned from genomics, is that we're going to have to get up into the sort of thousands to tens of thousands of people to really have the power, maybe even higher than that, as many as possible, to see some of these key differences there. But it was definitely very pleasing to see these number of pesticides in this sort of analysis. And so what lets you do is really start thinking about these sort of exposome-wide association studies. And because we're also measuring those biological pathways at the same time, like you're seeing elevations with this amitrin compound, but then alterations in different biological pathways that start giving you a clue of how these compounds might be interfering with our biological processes. And so I think this is where we've really wanted to be able to get to and being able to do this level of study. Also started a project a couple of years ago. This was a Cancer Grand Challenge project led by Melissa Davis at Morehouse School of Medicine in Atlanta, where she was very interested in looking at why people of African descent, whether in the UK or in the United States, have worse outcomes for cancer. And so, one, they wanted to do a lot more genomics on it, which is what usually people do. But they asked us to do an exposomic analysis in the same population. And so we're hoping to get thousands of samples from Kenya, Ethiopia, Nigeria, Zambia, South Africa, and perform this high resolution mass spectrometry analysis, but also have the tumor biology and have the genetics and be able to pull all these things together. Just as an initial check, when we started this project, I asked my team members how many of the IARC compounds could we detect, like all the IARC list of carcinogens, and it was like over 90%, like we could pick them up in our systems. And so, like, again, it shows you that we have this very comprehensive way of measuring how these small molecules are associated with various disease outcomes. Just a pilot study we did with them. This was a population, it's actually a Maryland, United States-based cohort of African Americans. But we were able to see differences between different molecules with prostate cancer. This was the one that we were studying here, but also being able to see difference between people of European or African descent. And so, like, we were giving these clues in these smaller studies, you're seeing the differences in the disease outcome, and you're seeing differences in the racial ethnic composition, which I think will start getting us closer to see why is it that you have worse outcomes in one population versus the other. Again, one of the things I really like about the approaches I showed, like with those lyso-PCs, is because we're not just measuring single biomarkers, like you're really measuring like families of biomarkers. And this was something we saw, this change in methyl lysine. It was different in the African Americans, but it's not like you're just seeing one of those molecules. You're seeing many different derivatives or metabolites of that pathway that are altered in the same direction. And so it gives you more confidence that you're not just seeing this single sort of biomarker, but you're seeing shifts in classes of molecules that, I think greatly increase the chance that it wasn't just a statistical anomaly that you saw that change. You're seeing really being things done at this sort of chemical class level. And I don't have time to go into too many of these examples here, but again, as a toxicologist by trade, like, I want to go and figure out how these things are doing this, like what are the mechanisms behind it. And so what we do is we use a lot of different animal models, but we do the high resolution mass spectrometry in those models as well. We've done it in cell culture, C. elegans, flies, zebrafish. And so what you can do is, if you see some sort of connection there, you can like expose those animals to the particular chemical. You can have a modified gene. For example, we use like a p-tau mutant worm, and we expose them to DDT, and then we can measure how that affects the outcomes there by using the mass spectrometry and doing behavior and other sort of whatever physiology you want to look at. But we've done this, looking at things like in manganese exposure in Drosophila, looking at things like with copper exposure in C. elegans, looking at some changes in like some dopamine neuron degeneration and mitochondrial function in C. elegans. And so the idea is you can use these same methods across multiple species, which I think is a very helpful way of like coming back and making these connections. Like when we talk about seeing a classic in lyso-PCs or something, you're not just, again, searching for one biomarker. You're saying we're seeing this pathway change, and what is that conserved pathway in another species? And so I think it's really going to help us bridge these gaps across biology. So we have a relatively new scientific agency in the US called ARPA-H. It's designed after our Defense Advanced Research Project Agency, but this one is based around health. And so they want really high risk, sort of aggressive projects. And so we proposed at one point was that, well, we could use these methods to see how the exposome affects how drugs work. And because we can detect a lot of drugs, why don't we just measure all the drugs? And we said, we'll take the top 100 FDA approved drugs and their metabolites, and we'll build them through a liver system to generate all the metabolites so we have them as controls, and then try to measure as many of these as possible in populations. And so it was a very kind of like daring sort of approach to say, like, let's just do this on a large scale, multiple institutions involved in it. And just like, short, so like this was like looking at one clinical trial for diabetes. And we had all their electronic health records. We knew what drugs they were prescribed. And then we could measure those drugs. And so like everything that they said they were taking, we could measure it. Like we could verify that they were on those medications. But we also saw in these clinical trials, many people that were prescribed the medication that didn't have any of the drug in their system. And so compliance is something we think about in various drug trials, but for some reason, we don't actually measure and check it. Like we can totally do this now because, again, you're not just looking for a single biomarker. You're looking at 4 or 5 metabolites to that drug. Like you really can see the patterns of people that are in a steady state on a medication. And then we even saw in many studies where there were people in the placebo groups that had the drug in their system. And so when you think about interpreting a clinical trial, when you have that level of misclassification, you're going to miss your targets. Like you're going to see things that you thought weren't significant, and it was just because the people weren't taking the drugs as they were supposed to. And so this is like a way of overcoming that. If we can measure the various chemicals that may interfere with metabolism, and thinking about, like we know the example of grapefruit juice where it interferes with cytochrome P450, like we can measure those molecules and P450 metabolites and see, what's happening and how all of these various external factors. And so I think we're really at this point of having pharmaco-exposomics that can merge with pharmacogenomics. And our goal is to be able to do this like at a biobank scale. Like, so we'll get about 10,000 samples done, but the goal is to have the system to be able to do like 100,000 samples or more. Like, so we can do something with like UK Biobank or all of a study in the US. So this Nexus project, hopefully everyone's heard about. So this was our coordinating center for the National Institutes of Health in the US. And so our goal here is to promote all things about the exposome. We're trying to help people with methods, study design, analytical things. And so we've just been really working hard on kind of building this infrastructure, developing these collaborative networks and developing educational material. Chirag Patel at Harvard and Rima Haber at University of Southern California are my co-PIs on it. We've built out this chemical biological sciences hub and thinking about harmonization trials. Again, while this is a US-based thing, we have opened it up to many of our European colleagues. We really want to do this as a global effort, but we still have like, in-country things that we need to do. But I think this is going to be very helpful in kind of building up the knowledge base and the ability for more people to integrate exposomics into their studies. We co-hosted a meeting with Irene back in October at the Mendel Museum. This was really a great meeting. What we wanted to do is bring like some of the top geneticists in to think about how we get to that G by E sort of approach. And I think this is going to be very critical because many of the large populations were designed for genetic type studies. And so we want access to those studies so we can get blood samples and do more exposomics. Ewan Burney was there. He's very enthusiastic about this. Kari Stephenson, who was the founder of Decode Genetics. When he came to the meeting, he didn't know what exposomics was, but at the end of it, he's like, they can't live without one another. Like you have to have the G and the E, and he definitely saw this idea that exposomics was the way to do the environment because it gets you a scale that's at a similar level to what we have for genomics. And so we'll continue having events like this to kind of, target different areas that we need to address in the field. And we're working on a manuscript now from this meeting on how to kind of advance this. Some examples from that meeting was this really exciting paper. Actually, Ewan Burney was on and presented some of this. Is how we're using some of these, generative transformers to do disease prediction, and how, again, like using these sort of AI-based approaches, we're getting much better predictive outcomes. And the more E part of it we get into it, these models will get even better. There were also like exciting papers, this one from Andrea Gana from using things in FinGen, showing how you could do like Mendelian randomization before you look at your environmental factors. It's like you randomize in advance because there's not a genetic effect of it, and it tightens up your noise in the data and you get better outcomes from it. And so like there's just, again, tools where we can learn from the geneticists and how they're approaching things to make what we're doing on exposomics stronger. We've seen Exposome a lot more in the news. We've been doing a lot of promotion again on our side, Nexus. We've started a podcast series, and we're willing to kind of get on and talk about the Exposome whenever we can. It's been really getting much better coverage in the news, both scientifically, but also things like in the New York Times and the Lancet and JAMA. And so I think it's a good sign that the field is growing, it's getting recognition, and we want to keep pressing it forward as best we can. So, it's a relatively short history, just about 20 years, but I think we have a very ambitious future. I think there's many things to come. I think we've built up a lot of the key, the foundational sort of work, but now it's time to scale and get to that level we need to have really strong outcomes from this. And I think that, again, I've mentioned this idea of exposome-wide association studies, but I think this is where we're ready for this now. So Chirag Patel has a paper in press in Nature Medicine that kind of lays this out very nicely. It'll come out in a few weeks. But this is something that we can do this now. The tools are there. We're ready to do this at a large scale, and it's time to do it. So this is what we've been proposing on the US side, like to the National Institutes of Health and ARPA-H, is that we think phase one of a human exposome project should be this large scale XWAS. So you think about like maybe around 100,000 people, you multi-omic, like, so in a lot of these studies, what's great is that they already have genomics done. I mean, they may have proteomics done. So come in with the high resolution mass spectrometry with the geospatial information and do a large scale XWAS across many different diseases, very similar to the Wellcome Trust paper in 2007 that kind of made GWAS work and scale it up. I think that's what we're ready to do here. Of course, there's many other things we want to do at Exposomics with, that's why we call this phase one. This is just like that first step, because I think this will give us the landscape of what's there and then figuring out where you have to get better measurements of exposures with monitors and things to do that. We still have ongoing challenges, is that we still need to improve our analytical methods. We lack the sort of infrastructure that that genomics and proteomics has had. Like, we don't have KEGG pathways for exposures, and we need those things. We need to figure out like those structures, and so we can have like a lattice on which to put the data to kind of make sense of these things. I think we need to keep exposomics anchored in fundamental biology. It's not just about describing associations. It's really understanding our physiology and pathophysiology. We think about the central dogma about the flow of information from DNA to protein. Exposome doesn't disrupt this at all, but it influences all of the components. And I think one of these challenges, though, is that we think about how a chemical might affect RNA or DNA and cause an epigenetic change. We're always doing it one molecule at a time, and that's not going to get us where we have to get. We have to embrace combinatorial approaches. And I think this is where proteomics will become a really key integrator to this, because it really becomes a functional readout of the genomic and exposomic processes. And so I think that we're getting there, but there's still many things to do. I think as we look at different sort of exposure matrices, we're seeing that many compounds do cluster together, which makes sense. Like if you have auto exhaust, there's a pattern you should see. And we have to start building out these systems to really advance the field. And so with that, just want to thank all the members of my team and collaborators all over the place for all the great work, and happy to take questions now or at the end or in the discussion period.
Thank you so much, Gary. It was really wonderful. We do have a few questions coming in, but please, colleagues, do feel free to raise your hand if you have a question. I'll take the first question, which is on how do you see the regulatory landscape evolving as exposomics moves towards more clinical and public health application?
So the regulatory area, this is an area that I will admit is not my area of expertise. I do think that having good human data is helpful. And I think the exposomics will give us better human data on exposures. And I think that will strengthen the case for doing this. But like, I think I look at what's been happening on the policy side in the US and thinking about regulating things like PFAS is that we regulate on individual molecules, and that's not how they're produced. That's not how we're exposed. It's not how they change our biology. I'd like to see more of a class-based system, but I don't know from the legal framework how you do that and how you enforce it. But I do think what it will influence is that we'll have better human data, human relevant data, and that it's very difficult when people say, well, rats are a little bit different, or like when you have human data showing these connections, and you get to the point of like, you'll still want to use some of these other animal models and things in here, or some of the, new alternative methodologies. But I think the key part is having strong human data to drive those decisions. Whether or not I don't know how you deal with the policy on the mixture aspect, though.
And maybe Alex or Gerardo could speak to it a bit from their experience as well.
Sure.
And there's a question a bit related here. So, what is your opinion on use of untargeted exposure measurements for risk assessment in highly occupationally exposed workers, such as firefighters or some others?
So I think that the non-targeted approaches are. They need to be used in the research, but when you're looking at like a clinical occupational sort of setting, I don't think the non-targeted approaches are going to be the best way of doing it. But what they'll do is help you identify the predictors of those exposures. Like ideally what you want, like we have now in many occupations where there's some sort of badge you wear, is I think that we'll have the technologies is use the non-targeted approaches to identify the 20 best markers that you want to look at, and then you put those on some sort of chip or with an aptamer or something like that. And that's the sort of thing you want to have in a workplace where you get an instant readout from it. You don't want to have to send it off to a lab to find out that someone had a bad exposure. You want real-time monitoring. So I think it's more on the research side of informing that risk assessment.
Yeah, thank you. And there's a question from Emma, which is on more, thank you so much for the talk. What role does gut microbiome play in exposomics, particularly in the metabolism of environmental contaminants, and how important is its inclusion in exposomics framework?
So I view it as being essential. I don't talk a lot about it, but because we're measuring things in plasma, it is downstream of the gut microbiome. So we do pick up many of these microbial metabolites. I think the first thing we're well aware of is that a healthy gut microbiome is a very good filter for chemicals that come into your body. So if you have a healthy microbiome, fewer chemicals and pesticides actually get into your bloodstream. So it's serving as a barrier, but as noted, it also can metabolize many of these different things. And so I think this is like where many of these unknown molecules we talk about are probably microbial metabolites that the gut microbes metabolize. You just don't have a standard for it, but it's just a breakdown product of one of the pesticides. So I think it's an essential part of it. And I think we need to have more studies that integrate some of the microbial species in with the exposomics. So yeah, very important, and it's a high priority for what we're doing now too.
Thank you. There are two additional questions. I think for the sake of time, we will try to take those two and then move on to the next one. So the question is on there are studies from UK Biobank where they first use exposome-wide analysis, then exposure with significant results are used in two-way Mendelian randomization with GWAS and an external GWAS. Is this an effective way to integrate environmental and genetic data, and should we first explore environmental exposures associated with an outcome and then verify their causal relationship, genetic variables?
Yeah, there were a lot of questions in that question.
In that one, yeah, that's true. It's a loaded one.
No, I mean, I think that I agree. I think that one, to me, like doing biobank level studies without actually taking advantage of the biobank samples, you need to have some of that data in there. But I agree. I think we need to have a kind of multifactorial approach. I think doing these things at a biobank scale and trying some of the Mendelian randomization is fine, but you also want to look, like you said, look at those direct outcomes where you have more causal associations. I think we're going to have to do many of these things in parallel. I don't have like the ideal study design to get to that G by E, but that's why we have to work and having those communities come together more to really come up with that. But I think we need to still have these multi-layered sort of things, not just biobanks. We need to have disease cohorts. We need to have, lifespan cohorts, child cohorts and things. We really need to pull all these things together.
Yeah. And then there's one last question, which is again more on the approach. So could you briefly comment on the causal inference approaches in exposomics research, where and how causality can be established, if at all, in this case? Yeah.
Yeah, I mean, this gets down, very much to the study design. Like you have to have, the natural experiments where people relocate or something, where you have that part where you can actually establish those causal relationships. When you're just doing things at a biobank level, it's very hard to get that if you don't have these interventional pieces. But for example, in things like in all of us biobank sort of studies, is that we have people coming into that study that are diagnosed with something like diabetes 1 month after enrollment, 2 months after enrollment, 3, like all the way out for years. And so in some of those studies, depending on what interventions may have occurred in them, you may have some of those clues to get to those causal associations. But I really think this is like where more of the natural experiments that occur from other epidemiological studies are going to be very critical, where those samples were collected where people had moved geographically or had different interventions for surgical intervention, but the blood samples are there. And so now doing exposomics on those samples that have the study design, I think that's going to be the most direct way to get to those causal associations.
Mm-hmm. And a bit of a final question from my side, Gary. Where do you see, for instance, eco-exposome or that complementing the human exposome, and where do you see it meeting in the One Health perspective? Like overall, where do you see that?
Yeah, I didn't even mention this at all because it's too much to cover. But to me, if we think about how we study biology, like we sequence the genome of everything in our ecosystem. Like we should be doing exposomics the same way. Like to me, genome and exposome should be part of all biology. And so it's a perfect fit into the One Health model, because if you, like when you think about an exposure, if you have a plant and you're getting exposure to a pesticide, well, it affected the plant too. And it affected the insect that was on the plant. Like we can do genomics on all of them, and there's really no reason we can't do exposomics across that to see how chemicals come up through the food chain or how various interventions reduce the exposures. And so I think One Health gets bigger and bigger. It can be mind-boggling, but it's like I think exposomics should be on the same level grounding as genomics is.
Thank you so much, Gary. It was wonderful listening to you. And then we move on to our next speaker. So Gerardo, we have who's with the EEA, and he's an environmental and health expert. So the presentation is the kind of exposome research we could use at the EEA, and probably it also bridges from some of the questions that we had in the last round a bit more in the regulatory phase. Gerardo, I give you the floor. If you could share your presentation, please.
Thank you. Can you see my screen?
Yes, not in the presentation. Yeah, now in presentation mode. Thank you.
Okay, thank you very much. I'm very happy to be here presenting today, and thanks a lot, Manoj, and all the colleagues. So I'm going to talk a little bit about, what we've been learning that is out there in terms of research on exposomics, and then how that could become something that we could start using. And I think it's good to start by saying that most of the regulations and most of the policy that the way it's designed today is not really designed to take these kind of things into account. So it's still at an earlier stage of how research could inform policy. But I think it's good to start by saying that we at the EEA are an independent EU agency, and the independent is an important component because it means that in theory we could be saying anything that is supported by data, even if this doesn't fit the narrative of politicians. I suppose that in the practice we need to navigate difficult compromises, but we like to believe we are fairly independent. We're based in Copenhagen. We analyze and assess data, and we very often talk to politicians, and we don't do it alone. We depend on very strong networks to carry out this work, and the strongest is IONET, which has, I think the number is outdated. I think it has well over 3,000 experts from 38 countries, and it includes also 7 scientific consortia, which along with our 300 employees put together all of this information and analysis to inform policies. We work on environment and health in groups with IONET. We have one on health and environment, but we also have some in air, noise, chemicals, and climate change adaptation. So we work with them, and the information that we publish is based on that, typically validated data from all of our member and cooperating countries. Why does exposome or exposomic research matter to what we do? So I think the first thing is that a lot of the chronic disease risk arises from modifiable factors. Environmental factors are still very significant in Europe, and yet it remains undermeasured and to a large extent undervalued, underestimated. So I think the case of chemicals is a very clear one. For that, Exposomics provides a comprehensive framework, and the comprehensive part is important because the way we're doing things now is more on a one by one substance case, and what we need to transfer towards is one where we take many exposures at the same time. But most importantly, of course, something that is preventive and driven by data is something that could help the EU ambitions on healthy populations, free of pollution, with safe chemicals, etc. What you see on the right-hand side is how we envision those main risks and factors. And this is from Europe's Environment Report of 2025. So I encourage you to have a look at this report. It's a comprehensive report we do every 5 years, and what we think and say about the environment and health is all written in there. Now, from a technical standpoint, there are quite a few things that Exposomics can help us with. One of them is the high resolution in terms of exposure surfaces. So of air pollution, of noise, of temperature, of greenness, and built and social environment, and particularly it'll be useful when it's at a very high resolution, so less than 100 by 100 square meters. And just to illustrate why it matters to us, most environmental inequalities that we can talk about now, or at least until recently, where we could only measure them at NUTS 3 levels, which for sorry, to illustrate how large that is like a large province kind of in most cases. In the case of Germany, they're quite small, but in other cases are much bigger. So the current status that we're struggling to downscale, and what you see there on the right-hand side is, I think for us, we're very proud about it because we're doing our best to cover all of Europe. And so this is a map of PM 2.5 levels at 1 by 1 kilometers for EU capitals, something that is available in our European Environment and Health Atlas. And this is the maximum level we are at right now. So in terms of where we would like to be is at a much higher resolution of at least 100 by 100 meters. So the problem that we'll have with that and inequalities is that even if we downscale in environmental data, we will not be able to downscale as much in terms of socioeconomic variables, but we'll keep on trying. Another advantage is, and I think this one is the one where we're seeing already the biggest advantages, is the internal exposome profiling. So targeted and non-targeted mass spectrometry, metabolomics, epigenomics, proteomics, etc., to map biological pathway perturbations. And it matters to us because ideally we'd like to see policy action occur before health outcomes manifest. Whenever we see chemical footprints of health impacts, say resulting from aircraft noise, we'd like to see that before it actually produces the health outcomes. Currently, we have no internal exposure or omics indicators, and we're even struggling to promote the idea of expanded biomonitoring of priority substances. Still, we are much better off today because after the HBM4 EU project became a much larger partnership for the assessment of the risk of chemicals project, which is what you're seeing there on the right-hand side, where monitoring and exposure, risk assessment, hazard assessment, and early warnings, it all comes together in a composite that makes sense for advising policy. Another advantage, and this one is crucial for us, is the idea that we could finally look at mixtures and cumulative effects, multi-exposure models, etc. Because so far we really are looking at things on a case-by-case basis, and that's really not how things work in the real world. But of course, we're limited by the capacity. We're probably severely underestimating chemical risk. I mean, the REACH and CLMS, which I've heard described, and I think rightly, as the most complex technical regulation on Earth, cannot possibly keep up with chemicals development. I mean, it's just impossible. So there's a tremendous burden of assessment. We're hoping that the one substance, one assessment regulation recently approved is expected to help. But in the practice, mixture effects are almost unknown and very little accounted for in policies. Now, another one is that with Exposomics, we can much better understand life course related impacts. And so particularly in sensitive windows like prenatal, childhood, working life, etc., and the elderly. It matters to us because this focus on sensitive population like children or the elderly is increasingly sharper focus, and there is a much better effect, I have to say, in terms of policy attention. Whenever we talk about children and also the elderly, we get a lot of attention that we don't get necessarily when we're talking about general population. Currently, many environmental thresholds are not firmly adjusted for most sensitive life stages. There are some exceptions in chemicals, but in general, they are not. What you see on the right-hand side is an example of our publications when we focus on children and adolescents. And this was one very good example of how the same exposure focused on a sensitive life stage like childhood and adolescence got a lot more attention, both from institutions and from politicians. So when we talked about general population, yeah, it was all fine, but it was generic. When we talked about children, we got like 20 times as much attention. Another thing that I think is important, at least it's important for me, is the integration of environmental and occupational exposures. So it matters to us because currently these exposures are seemingly disaggregated or like separate just because they happen to be in different administrative compartments or divisions. So there are separate exposure assessments, but in the real world, individuals are exposed to environmental risks at home, at work, at school, everywhere, right? And we need to integrate those because individuals are the same, and this is how things work in the real world. Yet it really remains separate in most cases, particularly low-level chronic exposure. So in terms of making it count, I think for me the issue is timeliness and following the file. So when you see this, there is this slide that I've used many times. I adapted it poorly from that particular website. I hope they pardon me. But you need to make sure that you're following a policy file and make sure that there's a window of opportunity there. And this is not something that is ethereal, like up in the air. Those policy of opportunities, those windows of opportunity are identified in most countries, in most regions, in most municipalities, and certainly in the European Union. They're very highly regulated, and it's clear when that the feedback can be put in. And it happens in many different ways. So through committees and through public calls for evidence, etc. So there's many opportunities to make that research count. Now, I encourage you to use every opportunity. So this in the left-hand side, you see extracts from the call of evidence on the now is not upcoming, is approved EU cardiovascular health plan. So many scientists would see a public call for evidence or feedback and say, well, who cares? They're not going to pay attention to it. Industry disagrees, and they always put their feedback, and they attach the evidence and peer-reviewed articles that benefit their case. So they don't think it's a waste of time, and neither do I. I would actually encourage you to subscribe to, at least in the case of the European Union, but also perhaps in your countries, subscribe to the Have Your Say website, which will keep you updated to all the policy files that interest you, including feedback, collections of feedback, and public consultations. I think that would be a great first step. So in summary, Exposome research is a very promising tool to inform environmental policy. There is specific technical advantages of Exposomics that can bridge gaps that so far are impossible to bridge with other types of research. So we should make it count in policy, particularly when we are getting punchier results. And for that, we need to identify the opportunity windows at all levels, particularly European, but also national and subnational. So that's all I have for today, but I'll be happy to take questions.
Thank you so much, Gerardo. I think there's one question, but something that you did touch upon it briefly, but I was more thinking about from your perspective, what would be more exposome-based markers that's robust enough to support your routine environmental biomonitoring and decision making. How does it look like in the agency so that people working in exposome research can kind of provide data in a way that, yeah.
Correct. Yeah, so say for example, one of the things that caught my attention recently in terms of exposome was when researchers were able to identify in animal models cardiovascular markers when they were specifically the animals were stressed by aircraft noise, right? And they were trying to identify similar markers to see how specific they could be in human populations. I mean, I know that's a far prospect, but something like that would be incredible to have because the idea of being able to compare populations based on those biomarkers and detect differences that could be related to environmental exposures, whether they be chemical or noise or air pollution, that would be fantastic, and it would make an incredible, a very compelling case in terms of moving the needle in policy. And so, just to illustrate also, those campaigns, it's just been essentially a little bit like publicity where figures, including politicians, would measure certain priority toxic chemicals in their blood. It's something that is impactful and also does move the needle in terms of bringing those discussions up in the policy agenda and in the relevant parliamentary committees. So it's just when we are able to get like clear correlations and correspondence between any omics marker and environmental exposures, if we're able to detect that more or less solidly in a population or a group of populations, and particularly if it's sensitive populations like the elderly or children, that is something that would help a lot. Certainly, that's a policy signal that would be very difficult to ignore for, say, European members of Parliament or the Commission.
Thank you so much, Gerardo. There's one more question, which is, if I get it correct, if it were only possible to look at one, two, three types of environmental exposure to help predict health consumption or health individually, what would you recommend to use? What are the most important vital environmental exposures to include in such predictions?
Yeah, I mean, I think right now what we need to look at carefully is so I'm going to expand to two. I think we need to understand well what is the true extent of the health impacts of exposure to environmental noise, because currently it is under-researched and underrepresented also in policy. So like the EU regulations about environmental noise are not aligned yet with the health-based thresholds of the WHO. So that is one. But I think the most important one is chemicals, where we really don't understand what the extent of the exposure is to toxic chemicals in the population and what the possible associated health burden could be, particularly as mixtures. So this is something that is really, every time we're asked, what can you say about that? And we had to say, unfortunately, not so much, particularly at the pan-European level. So having any of those exposures well covering most of the European Union or EU countries or EEA countries, that would be fantastic. So that's why for us, having something like expanded human biomonitoring would be a great thing to have. But even like putting together with one or two country studies per country, even if it wouldn't be necessarily a human biomonitoring campaign, would help a lot. Essentially, it would be about having enough coverage of key priority toxic chemicals. That would be great.
Thank you. And I think there's one additional question. So it says, I totally agree on the lack of information of occupational exposure in the exposome that may undermine the associations with outcomes. How can we approach this more effectively?
Yeah. So I think there are positive signs in that within the European Union. I can speak positively about the One Health approach, where in the practice, what it means. So I'm not going to go into whether the One Health approach is the right one or the wrong one. I mean, for me, as long as it does the job, it's fine, it's acceptable, right? I think it's also scientifically sound. So in our case, with the One Health approach, the main immediate advantage is that we're no longer having that conversation end there, which is, why are you doing this? This is beyond your competencies, right? With that's no longer happening, or at least not the way it used to happen. So now we have more freedom to engage directly. For instance, in our case with the EU Occupational Safety and Health Agency in Bilbao, right? So we routinely are exchanging data and information and reviewing and complementing each other's assessments. And this is already happening at the EU level. In many countries, the separation between occupational safe and healthy authorities and environment and health is like a brick wall. There's no communication whatsoever. So in that regard, I have to say that the EU is actually even a little bit advanced compared to many countries. So those walls need to be torn down, and there needs to be a pressure from scientists to say, listen, an environmental exposure cannot be complete in many cases without an occupational exposure assessment. It's as clear as that, right? It's not only for working populations, a lot of occupational exposures are sometimes inadvertently brought home as well by the workers, right? So all of those things need to be looked at, and I think we may be shocked at the magnitude of the effects that we might found with that.
Thank you, Gerardo. I think that's all questions we have. But if anybody has any other question, please feel free to drop. We can take it up after the presentation of Alex as well. Uh, Meanwhile, thank you, Gerardo, and I ask Alex to join. So Alex is the policy officer at DG RTD with the European Commission. And Alex's presentation would also look at EU-funded research and innovation on human exposome, looking within and beyond. So it would be nice to see how, and the domains of interest of Alex is environment, health, and equity nexus, chemical pollution. So would be able to bridge together some of the discussion that we had before as well. Alex, the floor is yours.
Thank you so much, Manasi. I will just share my presentation. Okay, I hope you can see my slides.
Yes.
Perfect. Okay, so thank you so much for inviting me to this symposium. It's a pleasure to be here. My name is Alex Mulet Indrayanti. I'm a policy officer at DG Research and Innovation of the European Commission. I'm not an environment and health scientific expert, but rather come more from the research and innovation policy side. Today, I want to give you a brief overview. Well, brief, it's a longer presentation than I'm actually used to making, but I will give you a rundown of the different activities that we are funding, that we have funded. Also give you a bit of policy context around this, since we've heard allusions to it, but maybe not in so much depth. And then also I will talk to you about upcoming opportunities and upcoming activities that we will be funding and where we're headed in the future. So first, let me start with some policy context. Okay. So policy context. So when we're talking about exposure and when we're talking about environmental degradation, pollution, these kinds of phenomena, we are starting to talk about lifetimes of exposure. This is, of course, not news to anybody in this symposium, but human life comes with a long and complex list of different exposures, chemical, biological, physical agents that directly influence health, and not to mention mixtures thereof as well. And of course, science helps a lot by actively uncovering these links between different exposures and non-communicable diseases and other kinds of conditions, including mental health conditions. But in some cases, evidence in regards to these links is early stages or not conclusive yet, meaning that a lot more science needs to be funded. This is a space that needs consistent investment. Of course, in Europe, efforts that have been undertaken to protect people's health from harmful exposures have advanced significantly, for example, through water treatment or clean air policies. But however, 87% of EU residents, particularly those in urban areas, still experience exposure to pollutants that are above the safe levels recommended by the World Health Organization. And this was a finding in the Environment Agency's Zero Pollution Monitoring and Outlook report of last year. So, okay, I'm gonna click instead of navigate with my arrows because I don't see the slides changing. Okay. In addition to that, the climate crisis also plays a crucial role. Climate change, of course, is a risk multiplier and exacerbates existing exposures, including air pollution, for example, which is the most notable case. It negatively impacts air pollution, and roughly 300,000 Europeans die prematurely every year due to poor air quality. There is also a clear increase that we have seen due to climate change in the incidence of climate-related vector, food, and waterborne diseases. And the kind of conclusion here is that adaptation measures are, of course, critical. They protect people, they prepare our populations and our systems from future harm, but they may not be enough to avoid new risks. So we will constantly need to adapt to new risks as time goes by, which means that climate mitigation, decarbonization, and a climate neutral society are really critical to secure our future in the long term. So these are the EU political priorities for this legislative term, which started in 2024 and will end in 2029. So sustainable prosperity and competitiveness, one of the priorities that are probably the most, let's say, communicated on. Security and defense, another example of that. Social fairness and strengthening societies is also a part of the priorities. Sustaining our quality of life by way of protecting our food systems, water systems, and also the natural environment. Democracy and values is another priority. Leveraging global partnerships, which is especially relevant in these current geopolitical times. And preparing our union for the future. So we're talking about preparedness, resilience, and adaptation, as I was just talking about. And hopefully, as I proceed with my talk, you will see kind of how Exposome ties in with these different political priorities. These are some recent EU policy developments that have taken place and that are aimed to protect environment and health. So these are just 5 examples. Of course, there's more, but I wanted to highlight here these 5. So the first is the CLP, so Classification, Labeling, and Packaging Regulation, which was revised in 2024, so already more than a year ago. And it was revised to include more simplified labeling, clearer information on chemical hazard for products that are sold online, and also rules to classify complex substances, which is super important given the sheer rate of chemical production in Europe. So that's the first example. Another example is the Air Quality Directive, which was also revised in 2024, and it has imposed stricter limits on particulate matter 2.5 and also nitrogen dioxide. It's also introduced regular review requirements for air quality standards and the possibility of compensation for negative health effects from poor air quality. The third one that I wanted to highlight is the soil monitoring law, which is part of the soil strategy as well. This one is from 2025 and aims to make strides in soil health, of course, forcing member states to monitor soil health regularly, also aiming to help farmers and soil managers to improve soil health and resilience, and also addressing the ongoing issue of contaminated sites. Then we have the packaging waste regulation, also from 2025, which is part of the plastics strategy, as well as the circular economy action plan, which placed restrictions on single-use plastics across various sectors, and also aims to minimize substances of concern used in packaging, including PFAS, as well as decrease the use of virgin materials overall in packaging materials. And finally, this was alluded to in some of the other presentations, the One Substance, One Assessment policy package, which was adopted just in January of this year, last month. and which aims to achieve stronger cooperation on chemical assessment in the EU across regulatory bodies, and also establishes a common data platform on chemicals and a framework for emerging chemical risks. So how does the research that we fund under Horizon Europe, which is our main instrument for funding research and innovation, how does this research and innovation contribute? So this is a pie chart that shows you kind of the relative policy contributions of the different projects and clusters that we have under our remit. And you can see that our research portfolio feeds into a wide range of policies. 45% of the policies that our clusters contribute to are environment policies, namely the Zero Pollution Action Plan, part of which includes some of these policies that I just highlighted in the last slide. The biodiversity strategy, the ambient air quality directive, plastic strategy, circular economy, etc. We also have 25% of the policies that our projects contribute. So the second biggest chunk is health policies. For example, the Drinking Water Directive, the Occupational Health and Safety Framework, the Beating Cancer Plan, also very notable. One Health, which is now a very hot topic in EU policy circles. The Mission on Cancer, and others. You can see the full list there. Then we also have climate policy, which we do consider separate for the purposes of taking stock, but of course is very much related to both of the two categories I just highlighted. And that accounts for 10%, so the third biggest chunk. And then we have successively smaller chunks dedicated to things like urban policy, animal welfare, life sciences with the life sciences strategy, which I will talk about a little bit later. Preparedness with the Preparedness Union strategy, and of course, social inequalities with the pillar of social rights. So you can see that we feed into a wide range of policies overall. And in terms of funding, historically, in the last 20 years, the EU has funded around 3 billion euros in environment, climate, and health research. And you can see here in this bar graph that over the years, it has, of course, increased. The most substantial increase being accounted for in the transition between Horizon 2020 and Horizon Europe. Horizon 2020 already saw a substantive increase in funding for this area of research. With Horizon Europe, we've taken it even further, far surpassing the numbers under Horizon 2020. This is kind of a breakdown by environmental risk factors of the different projects that we have had funded under Horizon 2020 and Horizon Europe together. You will see in the pie chart the share of funding with also the figures. And you will see that overall, the largest area that has been funded has been chemical risks, and it has been for a while. We're seeing now different fields kind of increasing the investment that they receive, namely combined exposures, which Exposome definitely falls under, which you see as the second biggest chunk of the pie chart. And then, for example, air quality is also a growing chunk of the pie. I think with the indoor air quality cluster and indoor air quality and health cluster, this has also had a significant boost. I just want to highlight also the climate change portion of the chart, which is now sits at 8.5% overall. This has seen a significant increase, especially within Horizon Europe, which I will also address a little bit further. And then the bubbles on the right-hand side of the slide are the number of projects. So again, you can see that chemicals projects are dominating our portfolio for now. This is a list of the different clusters that we have. So under the environment, climate, and health portfolio, what we do is we ask projects selected under the same call to form a cluster, which means that it's a feedback to policy tool and a tool to identify synergies between projects to work together on different aspects like communication and dissemination, data and data management, and of course, science for policy. And the idea is that they collaborate despite their differences to find common approaches and common synergies within the cluster and enrich the different research groups involved in the different consortia. So it's really a fantastic instrument that has given us a lot of fruit, and which has also been kind of appreciated by other policymakers at the EU level and also beyond the EU level. The clusters produce things like policy briefs, newsletters, brochures, joint publications, etc. So I will not get into it just in the interest of time, but I want to mention, of course, EHEN, which is the first one that you see on the chart, which is the European Human Exposome Network, which came to an end last year. I will talk a little bit more about it later. We also see Expo Health Net, which is the last one of the chart, which of course is much more recent, and EXPOSIM belongs to this cluster. So I want to maybe give a few examples of EU-funded Exposome research from the past, which I've also seen in the earlier presentations. In Gary's presentation, for example, I saw this initiative alluded to. So that is the EU Exposome initiative, which took place during 2012 and 2018, funded under the 7th Framework Program. So just one program back from Horizon 2020. With a total of 29 million euros. So back then we were talking about much inferior figures, and a total of 3 projects were funded under this initiative. Exposomics was the first one, which used perturbation of adverse outcome pathways as a conceptual framework, and developed this framework for systematic measurement of exposure, including identification of biomarkers, pooling and integrating information from both short and long-term experiments, and also evaluated health outcomes and physiological change as a result of exposures. Then the HEALS project looked at environment-wide association studies, of course, in support of EU-wide environment and health assessments to understand the development of major non-communicable diseases. It also worked to advance data integration, including integration of environmental, socioeconomic, exposure, biomarker, and health effect data, as well as bioinformatics. And it explored the causality between exposures and risk of disease by relating omics and prenatal exposure and early life health outcomes. So it focused on early life. And then we have the Helix project, which also focused on early life. It built a early life exposome database and elucidated correlations and determinants of the exposome, of the personal exposome, including, of course, complex correlations, and also evaluated risks related to multiple exposures, which, was also important. This was by no means the first exposome projects. These were not by no means the first exposome projects that were funded by the EU, but it was certainly the first notable initiative that we can mention. In the successor program to FP7, we have, of course, under Horizon 2020, the European Human Exposome Network, EHEN, funded with 105 million euros, so substantially more investment. It comprised of 9 projects. I see the link to the website that just popped up in the chat. And the main outcomes of the EHEN cluster, in my opinion, were the Exposome Toolbox, which is a hub that was put together by the cluster for tools and resources developed by the different projects. And here I will mention just 2 examples that I remember, which are a tool for conducting occupational exposure assessments in large populations, Eurogem 20, and also a pipeline called My Purchases Pipeline for matching, validating, and enriching consumer purchase data with health data. So those are just 2 examples. There are many more tools in there. These tools are now being picked up by, kind of successor initiatives that have transferred them, and maybe they will be able to take them further. The other thing that I think is really notable concerning EHEN is that they have really advanced research data approaches to study environmental risk factors and the relationship to health. I'm thinking, for example, of the 6SAFES model that was developed to reduce the risk of re-identifying data subjects in regards to sensitive health data. And also their contribution to the European Health Research Data and Sample Catalog, which of course is a searchable tool for metadata that kind of allows combining and analyzing different sources of data. Another thing that I wanted to mention is I'm not intimately familiar with EHEN, as I didn't directly oversee it, but I know that we ask our clusters to produce data management plans. This is certainly a requirement under Horizon Europe, and the idea behind this is for the projects to find synergies in regards to research data and data management, to share best practices, and potentially, if there's room for it, develop common approaches to data. And I've heard in several meetings and several panels and fora that this is something that really helps the cluster to kind of advance the field in terms of their respective fields in terms of data. And that it's much appreciated, this kind of pressure from the Commission to build something together in regards to data. And then the last thing I wanted to highlight in regards to EHEN is their fantastic policy brief that was published, I think, not too long before the cluster lifetime came to an end. And this policy brief contains a lot of rich information, but specifically policy recommendations and calls to action aimed at decision makers, which is, again, one of the objectives of these clusters is science to policy. I also want to highlight IRENE, which was also mentioned in Gary's presentation, which is, of course, the European Environmental Exposure Assessment Research Infrastructure. The preparatory phase of this research infrastructure was finished last year, and it was funded with 2.9 million euros under Horizon Europe by a coordination and support action. This research infrastructure addresses the need for having a centralized source of data on chemical safety, pollution, and health impacts. It kind of fills the gap in terms of consolidating capacities, tools, and data sets that were developed in other EU-funded initiatives, for example, HBM4EU, which was also mentioned today. It also aims to support the development of technologies and integrated services to characterize complex exposures as well as their effects. And this is also work that is underway in many different projects, and also under the PARC partnership, the Partnership for the Assessment of Risks from Chemicals. So, you know, IRENE really looks to be that kind of glue and to provide the services that will link these different initiatives. And now the implementation phase will begin. It began as of 2026, so this year. And the idea is to transition towards an operational stage as an ERIC infrastructure from 2030. Then I wanted to highlight the International Human Exposome Network. So this was funded under Horizon Europe as well, with a budget of 3 million euros, another coordination and support action. And as we've seen, Exposome research, I mean, at least in the EU, has picked up a lot of pace. It's become rather popular with a lot of different projects popping up under the different framework programs. This, of course, has inevitably led to an increase in outputs, an increase in publications of data as well. And what IHAN seeks to do is centralize these efforts and foster kind of a structured cooperation at the international level, with the ultimate objective of advancing Exposome, the Exposome field or Exposomics field, in the best and most effective way possible. As I mentioned, well, I didn't mention IHAN specifically, but when I was talking about some of the tools in the EHEN toolbox being transferred to other initiatives, IHAN is what I was referring to. So there's also the IHAN toolbox that currently features some of these EHEN tools. And something that I just wanted to say is that initiatives like IHAN are really important because they chart the path forward for the future of the field. And in the current geopolitical situation that we're living in, we're seeing multilateralism and international cooperation kind of be threatened, and we need to stay in the fight. We need to, unite, make sure that we unite people that are like-minded and that we bring these like-minded partners to the table. That's why we want to keep funding initiatives like IHAN, which kind of try to harbor international support to pursue these very important research questions, and that will ultimately contribute to the protection of public health in the face of the planetary crisis that we face. Okay, just briefly, because I know that this is an Exposim symposium, I will not bother you with the details of your own project, but there are two projects under Expo HealthNet that focus on Exposome. Exposim is one of them, looking at immune-mediated diseases, and the other one is Exposome, which looks at the mechanistic role of different kinds of pollution in the development of non-communicable diseases. And the kind of most notable features of the call back in the day were to look at, the associations between pollution and disease, which is something that we heard a couple times today, that is necessary to look at, and of course the causal mechanisms and the biological pathways. And if that's done across the life stages, then that kind of captures the essence of what we're trying to do with exposome research. So just to very briefly say, this is the cluster that is funded by a lot more than 60 million, but 60 million refers to just these two projects that are working on exposome research. Okay, so I've given you a rundown of the things that we have been funding, kind of, the activities that the most recent activities that we have invested in. And now I want to look a little bit beyond, and I want to look at towards what we're headed and what is next in Horizon Europe, but also beyond Horizon Europe. So the first thing that I will mention is we have a new work program in Horizon Europe, which I'm sure you're aware of, or some of you at least are aware of that. It was adopted back in December, and under the health work program, or the health part of the work program, we have Destination 2, which is fully dedicated to research on environment, climate, and health. And in the work program of 2026 and 2027, the published calls indeed focus on understanding and addressing climate and health issues. But of course, that doesn't mean that there are not topics relevant to Exposome researchers. There very much is. In the 2026 work program, which is the first three topics that you see on the screen, we have a topic on better understanding and anticipating health impacts of climate change, whether it's in relation to non-communicable diseases, mental health, or infectious diseases. Then we have a topic on building climate resilient and carbon neutral populations and health systems. And then finally, we have a coordination and support action to provide support for a multilateral initiative on climate and health research. Then in the 2027 work program, the bottom two topics, there is a topic focused on the exposome. So this topic tries to increase, or aims to increase, the integration of climate-related exposures into exposome research, which we identified as a gap, as well as characterize the changes that take place in the exposome as a result of climate change. So how the exposome changes with climate change. And as I said, the risks are emerging, the risks are changing. So this is also a very big gap. And the last thing that we have in the work program for 2027 is a pre-commercial procurement topic. This is a new instrument that we're experimenting with in the environment, climate, and health portfolio. We have never done this kind of instrument before. We've never funded this kind of instrument before. And this one will focus on the development of tools and technologies for health adaptation to climate change. If you would like to learn more about any of these topics, you have all the links here in the screen. I think you can even click on them because I'm sharing the file directly, so feel free to do that already. I'm sure you will also receive them later on. And I also invite you to join us for the Info Day, which will take place next week online, 10th of February, 9 to 5 p.m. Central European Time, and the focus of which will be the 2026 topics. So if you're interested in learning about first three topics, I very much encourage you to sign up. It's a great opportunity where you can pose your questions directly to the policy officers that have written these call topics. And just to mention that if you do come to the info day and your question is submitted, you submit a question and the question is not taken up, that doesn't mean it will not get answered. All questions get published on the funders and tenders portal after the info day. So fret not, you will have your question answered. I want to briefly talk about this strategy for European life sciences, because our future activities will also very much be shaped by this strategy, which was adopted in July of last year, and which is very much about competitiveness in the life sciences sector. The objective of the strategy is to position the EU as the most attractive region in the world for the life sciences by 2030. So clock is ticking. So the strategy is kind of a way to help build a sustainable economy in the European Union and to secure our economic security, if you will, and kind of gain that independence from other large economies and yeah. Then just briefly want to maybe introduce a little bit why I'm talking about life sciences and why life sciences is important, because maybe we're used to hearing about life sciences in a different context. But the life sciences strategy, what it does is it adopts a holistic view, a comprehensive view of the life sciences. And what I mean by that is a view of life sciences that covers many scales. So cell to organ to organism to, to ecosystem to planet, that kind of scale. And it recognizes that the life sciences sector really has the potential to deliver knowledge and products in a range of very different fields, including health, including food, agriculture, materials, and also environment. And excuse me, through its flagship actions, the strategy also acknowledges the interlinkages between these different sectors and also between the different subsectors. So, for example, that whatever affects health affects environment and vice versa. That's an example. The life sciences strategy does a really good job at capturing that kind of interconnectedness. And I want to highlight just three of these of the flagships that are under the life sciences strategy and one non-flagship action that would be potentially relevant to disseminate here. The first one is the flagship on One Health. In particular, the ramping up of the Commission's activities to promote the One Health approach and its operationalization in research and innovation, of course, where beneficial. And of course, as we've heard, One Health and Exposome are very complementary approaches, and they kind of feed into each other. And the breadth of One Health, which in more recent years has kind of gone beyond just human, animal, and ecosystem health, but also, increasingly including other health influencing factors, let's say. This approach can really benefit from evidence that is provided by exposomic studies that kind of look at the collection of all exposures in a lifetime. So the flagship on One Health looks to identify priority areas in research and innovation that would benefit from One Health approaches. And it will mean that there will be guidance developed to support inter and transdisciplinary research and innovation in One Health. And for this, nearly 100, sorry, not for this, for to transform the EU into a world-class innovator in One Health microbiome-based solutions, there will be a mobilization of nearly 100 million euros under Horizon Europe. And then the second flagship I wanted to or actually this is the third flagship, forgive me, because there's two flagships on One Health and one on climate change and health. So the third one is on climate change and health and consists of the mobilization of 170 plus million euros under Horizon Europe for research on climate change and health. And this is to implement the Strategic Research and Innovation Agenda on Health and Climate Change, which was published last year in June. You have the link there on the screen if you're interested in reading it. And the other part of this flagship is the proposal by the Commission to set up a platform for global cooperation between funders in the climate health nexus, which will hopefully foster alignment between funders and support the development of solutions to climate and health issues, both in respect to adaptation and mitigation, of course, always looking at the health context. There's also an action on the future of food systems, which I wanted to highlight because it might be relevant. And it aims to develop a strategic research and innovation agenda, this time for food systems. And this is to foster kind of a competitive, sustainable, and resilient food systems in Europe and beyond, I guess, because they're all interconnected. And this RIA will also complement the strategic approach to research and innovation in agriculture and forestry, which was already announced in the vision for agriculture and food. And with that, I will give you a little bit of information about what comes after Horizon Europe. So in other words, FP10, which for now at least has been dubbed Horizon Europe Part 2, which is either very uncreative or very smart, depending on what you think. Under the structure of this new Horizon Europe that has been proposed, because it's still under negotiation, the structure looks a little bit different than the current framework program. So there's still the 3 pillars from the old program, but they've been slightly modified. The budget for the innovation pillar has been substantially increased in the interest of harboring competitiveness. And there is also now a dedicated pillar for the European research area and activities under the ERA, including research and technology infrastructures. So the research that funds EXPOSIM and other can you hear me?
Yes.
Yes.
I'm not sure if we lost Alex. His screen seems to be frozen. Well, I had a question for Alex. I think he'll join back, but maybe a question.
Back in.
Yeah. Let's see.
I am so sorry, I got disconnected. Something happened with my Wi-Fi. Can you hear me now?
Yes, we can.
Okay, so sorry about that. Let me just pick up the presentation where I was. Resume. Okay. Okay, sorry about that again. So, as I was saying, the research that funds EXPOSIM and other projects in environment and health is proposed to still be funded under Pillar 2, which will this time have 2 portions. So 2 kind of sections, one for competitiveness and the other one focused on societal issues. The competitiveness portion will be subdivided into 4 policy windows, which correspond to the same 4 policy windows of the upcoming European Competitiveness Fund. And the relevant window for us let me think, I think I have an animation. Yeah. The relevant window for us is the health, biotech, agriculture, and bioeconomy window, which will enable health research to contribute to priorities various priorities, including, for example, decarbonizing health, securing value chains, making sure that people stay healthy and productive, and of course, developing technologies to tackle climate and health issues. Also, just to mention that the New Horizon Europe will be closely linked to the Competitiveness Fund that I just mentioned, which is going to hopefully facilitate a lot of the uptake of technologies and products that are developed and piloted under Horizon Europe, which gives answer to issues of sustainability of outputs that have been there in the past and that have been raised in the past. And just concluding here my presentation, just to give you an indication of what has been proposed, fingers crossed that the negotiations have a good result. The health policy window is proposed to have 19.7 billion euros in budget, which is a substantial budget and the second largest under the competitiveness pillar. So if negotiations yield a good result, as I said, then we will have substantial resources for health research in the new program, and that includes, of course, environment and health research. So we're very happy about that. And with that, I'd like to close my presentation and invite you to visit our website if you would like to learn more about our activities, the projects that we fund, and the clusters that we have under our remit. Our fact sheet also gives you a good idea of that if you want kind of a more digestible format. And then we have a catalog of environment and health projects from Horizon Europe. You can see a comprehensive list there of what we've been funding. Thank you so much.
Thank you, Alex. I don't see a lot of questions in the chat box, but I do have a question, and I think you did touch upon it, and it's a question also for probably Gary and Gerardo. In terms of sustainability of a lot of these tools and rather integration, because I know Gary and colleagues from other European partner institutes are working towards many of these. Could you speak to the sustainability and more harmonization and how that ultimately integrates into the European health data space? Because that's another aspect that we probably need to reflect on a bit when we are generating more data in this Horizon Europe project, but going forward. So
I think in regards to the EHDS, maybe I'll address
Alex,
that part
just
first.
a minute. We don't see you a bit, but
you don't see me?
Your camera is off. Yeah.
Oh, okay, wait, do you see me now?
No. Yeah, now yes.
Okay. So maybe in regards to the EHDS, we have an infrastructures work program which we don't oversee ourselves, but it's an integral part of Horizon Europe. It's the work program that has funded the IRENE preparatory phase. So there are initiatives like IRENE that are kind of being funded to facilitate the bridges between the different actors in terms of data. And what I can say is that once these research infrastructures and other initiatives, the databases, biobanks, whatever, once they have like a legal entity, they can then start operating and they can become an authorized participant of the European health data space. So the options are there. I'm not an expert on EHDS. I don't know what the specific pathways to do that is, but we are hoping that the EHDS, because it's such a comprehensive regulation, and of course it takes into account environment and health data as well, because that's also health data, that this will be the path forward. And that's why we mentioned EHDS in our calls, in our latest calls, and going forward, we will continue to do so. In terms of the sustainability of outputs, for now, what has been proposed is the Competitiveness Fund, and that looks specifically at technologies and products, if I'm not mistaken. So that kind of tackles the issue of kind of technologies that don't get picked up after the lifetimes of the projects. Hopefully the negotiations go well, and it means that we have an instrument to do that in the end. That's the idea of the proposal. But it's true that then there's other types of outputs, maybe, for example, dissemination work, communication work, things like this, that are not technologies. They're not kind of tangible physical materials that maybe also need to be kind of helped even after the lifetime of the project. But hopefully, we will see that in the end result of what FP10 ends up looking like. Maybe I give the word to the others because I see we're running out of time.
Yeah, and a quick question. I don't know if that's something, but when you say about the sustainability through the competitive fund, is this something that's a bit of a continuation of the innovation radar, or a bit? But maybe we can listen to it in the info day and other sessions as well. Maybe the other speakers.
Yeah, you can. Well, I'm not too sure about the innovation radar. It's the European Competitiveness Fund is a separate instrument. It's not a Horizon Europe initiative, and it's not a kind of a research and innovation policy, if you will, or instrument. But it will be closely linked to Horizon Europe, so such that the research feeds into then what makes it into the market in a more direct way that is kind of curated and kind of overseen by the commission. So yeah, to answer your question, it's a new instrument which builds on many things, and one of those things that it will build on is Horizon Europe.
Thank you. And if Gary or Gerardo would like to add something, please go ahead.
But I think that the data storage sort of issues and the tools, I mean, this is all branches of science deal with this, right? Like you have projects that they're supporting these systems. And so I don't think there's anything unique about it. I think for the field, what's critical is to make sure that the tools that are useful remain useful in some form. And I think this is where we need to look at multiple ways of doing this, because it's not just sustaining it on that one project. Like, for example, we talked quite a bit with EMBL, EBI on getting more Exposomics tools like into their workflow and their system, because they have a long-term sustainability plan. But I do think it's something we should work hard on across different countries too, because we need to have these platforms and these tools available. And I think having the next phase of Irene is very helpful, because that gives us more of a track forward on that side. But it's still like, how do you move the key tools around that were developed under one project into the next system? And fortunately, most of these are all open source things that allow us to do that. But we really have to be proactive to make sure that these useful tools continue to be accessible.
Thank you, Gary.
Yeah. So on my side, just want to say a couple of things. I think the EU has become a lot better at trying to think about the sustainability of what's produced under EU-funded projects beyond the lifetime of those projects. But I think it's still there's a ways to go in terms of sometimes it's not necessarily the pure innovation that is needed, particularly when it comes to environment and health related research, but maybe the coverage that sort of becomes relevant for the policymakers and institutions, particularly at the national and subnational level. And then in that regard, I think it needs to be absolutely clear to the audience that we cannot possibly be thinking that the sustainability of the omics research in Europe could come from EU funding. That is just not realistic. It's a very naive and unrealistic and even dangerous way to think. I think the funding needs to come from a multiplicity of sources, and in eminently preventive fields like this one, there needs to be, I think, a healthy balance of public funding and also private funding to the extent that is possible.
Thank you, Gerardo. With this, I think we are almost out of time, but thank you to the speakers. So Alex, Gerardo, and Gary for being here with us and answering the questions. And I want to acknowledge the consortium as well, both the Exposim consortium, which you can see on the screen, and also the Expo Health Net Cluster, which is trying to proactively work in the field and move the needle a bit forward. If you want to reach out to us, please do so at any given time. And I think there would be a survey shared at the end of the symposium on the chat, but also by mail. Do put in your feedback, your comments. that always helps us to improve. So we will try to do that, and see you in the next symposium, which will be announced in a couple of months, I suppose. Thank you.
Thank you so much.
Thank you.